get her started. Thursday's kind of like our Friday, right? So, almost there to the weekend. We had about 5 or 6 slides, I believe, that we didn't finish in the last time, and so I want to start with those and continue into this world of the nervous system and how the neurons interact and are involved with creating memories and things like that. And that's going to be our big topic on Tuesday's lecture next week. So today we're going to finish up and then get into the types of communications that the synapses are involved with and talk a little bit about neurotransmitters. I think today's lecture is, so if you don't have lab, then party on. But every time I say it's a short one, it never is. So sometimes I'll say, we're going to get out like 20 minutes early. It never happens.
Quit jinxing us.
I think it could happen today. So last lecture, we talked about a pathway, and we drew a pretty impressive pathway on the board. Much more complicated than what we see here, but we talked about the fact that it always begins with a stimulus that some type of receptor is identifying. We said the receptors could be identifying external stimuli or internal stimuli. We use the words somatic, and we use the words visceral, and we use terms like sensory and motor and afferent. Oh my gosh, last lecture was a pretty big one. And so when you look at this simplistic view of the pathway, you can see that they do utilize some of the terminology like which type of neurons are involved. And we call the spinal cord, just like we did in lab, an information highway in which information travels up and down through the spinal cord. The ultimate response, we said in class on Tuesday, was that we have a result or an effect, and we call the things that do the effect effectors. And we said muscles and glands are usually the end result. Sometimes they're visceral, which means the effect is something that we're unaware of. And sometimes they are somatic, which usually respond or reflect skeletal muscle movement. We drew a neuron on the board, and we talked about dendrites, the cell body. We talked about the trigger zone, remember that? And we talked about the axon being wrapped either by oligodendrocytes or Schwann cells, and the fact that we have these little open gaps that we tried to see and identify in lab, called the nodes of Ranvier. At the very bottom of the tail, you can see that there are some synaptic knobs, which we're very much comfortable with, I think, since we've been talking about that with neuromuscular junctions, so that was good. And then we progressed into something that I thought was kind of a review from the world of muscles, and we talked about action potentials again. And we said that instead of the sarcolemma, we're talking now about the membrane of the nerve cell. Sometimes it's called the neural lemma. It's the same thing. It has channels, and it has a more positive outside ECF and a more negative internal cellular fluid. So when we have abrupt changes, because sodium, a positive cation, is rushing in and potassium leaves, we still get that same result, a spark, which we call an action potential back in the days of the sliding filament theory, and we're calling it an action potential in describing how a nerve signal is set. So an action potential is the electrical signal that's sent down an axon. This was the slide that we ended with. I said that this was photo worthy, and I said that you should be able to look at this and explain all parts of this diagram. We started off by saying that at rest, the membrane is considered very negatively charged, and that when sodium rushes in, we get to polarization, but only if we have enough local potentials, remember those, that accumulate at the trigger zone. So if that reaches threshold, the sodium floodgates open, then the potassium floodgates open, and we get a spark. That's what this spike represents. And the last thing I said on Tuesday is I mentioned that there is a brief dip below resting membrane potential, and we call this hyperpolarization. And during hyperpolarization, it is impossible, well, it's very hard for another impulse to be sent at the same time. This is something that's different. I think that something that is unique to a nerve action potential versus a muscular action potential. If you remember the word tetanus in muscles, a tetanus muscle is when the muscle is contracting maximally and not resting. I describe it as being like a Charlie horse. You know, you're having a lot of signals sent at once. You don't get that with brain activity. You don't get a massive amount of signal being sent at once. You get one response at a time. So hyperpolarization allows for a brief period, even though it's tenths of a second, where there's a break. There's a little bit of a chance for it to recover. Okay, so that's where we're going to pick up and finish up the last 4 or 5 slides from last class, and then we'll get into the world of neurotransmitters and synapses today. So this slide and this slide are the same thing, believe it or not, just it's scrunched. The time interval is scrunched. And what you can see is that when we get a depolarization followed by a repolarization, we get something called a spike. And this is really what causes an electrical stimuli to be sent. We said that an action potential is different from a local potential. Local potentials, we said, are weak, are decremental, can be reversed, and none of that applies to an action potential. An action potential is a massive signal that's going to be sent. It's not going to get any weaker, and it's not reversible. So when we look at the slide of the graph that I told you was really important, you can see that there's an area that's highlighted in the area where it dips into this hyperpolarization kind of situation. And during that time, we say that it is, look, relatively impossible to get another action potential. Well, I guess it's somewhat or mathematically possible if we have some incredible stimulus that gets us up to the threshold, but it's really hard. And notice that during the actual action potential itself, we're in a period that says that there is absolutely no way that you can get 2 in a row, or 2 back to back, or 2 at the same time. So if you combine both of these colors, you can see that it provides an opportunity for one spark to be created with a brief amount of time of rest before another one. So there's no such thing as tetanus in the world of nerves. Okay, that's good. Now let's talk about how that nerve signal travels. And some of you should already be thinking, oh, I already know how it travels down the axon. It travels very quickly if it is myelinated, where it travels slow and steady if it is unmyelinated. Remember, the purpose of myelin is to speed up the transmission of this nerve signal, this action potential. So let's start with an unmyelinated axon. That's what we see right here. So this is an unmyelinated axon, which I told you there are some examples in the human body where we need slow and steady responses. So what you see here is an axon that looks very similar to me as the membrane of a muscle cell. So you can think of an unmyelinated axon almost as if it is the sarcolemma. There are tons of voltage-gated channels sitting side by side by side by side. And if we get one experience where we have a depolarization followed by a repolarization, that spark is enough to open up the next voltage-gated channel right next to it. And then that one opens up the one next to it. So the movement of a nerve signal is exactly the same that we saw traveling down the sarcolemma until it got to a T2 joint. I described it as a line of dominoes going tick, 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 tick. That's not very fast. I mean, it's fast, but it's not as fast as if there was myelin around it. So a unmyelinated nerve fiber is slow and steady because we now know that it has these voltage-gated channels back to back to back, creating kind of this slow and steady response. Okay, that's good. So what's the difference and what's the big deal with myelin? Well, we know, look, that this axon, right here where I'm touching, has a lot of voltage-gated channels. It's the same thing as this. But there are regions where we either have Schwann cells or oligodendrocytes wrapping their wrapping their myelin around this axon. And what we have is we have an incredible amount of sodium rushing in these spots called the nodes of Ranvier. And what happens is that we have a tremendous amount of sodium rushing in a concentrated area, and this causes a signal to literally, listen, jump to the next node of Ranvier. Because it's covered, right? So the signal jumps, it opens up a bunch of floodgates, we have a ton of sodium rushing in, potassium rushing out, it jumps to the next. So what we see is that at the nodes of Ranvier, and the purpose of the nodes of Ranvier is that we get lots of salt, sodium rushing in. And it causes this signal to jump from node to node much faster than it would if it was causing individual voltage-gated channels to open and close in a row. So it says no sodium can enter, or no potassium can enter or exit where there's myelin, but in these nodes of Ranvier, it's very concentrated. And that seems to be the explanation of why myelinated axons are faster, is because we have this signal jumping to each one. So that's the explanation, and that's the kind of the reason why myelin is so important, is that it's covering up really these voltage-gated channels and allowing for an incredible burst to happen in these nodes. So here's kind of a diagram, don't take a picture of this, but you can see that we have voltage-gated channels in the nodes of Ranvier, and they open and they send the signal and it jumps to the next one and jumps to the next one. So there you go. Okay, so that's good. Now we get to move into today's lecture. And I'm going to tell you a little bit of a story that's not on your outline, but a story that I will put on the final exam. So listen to the story, make sure you understand the point of the story. And I will title this story Cage Potatoes. Okay? That's the name of this story. Cage potatoes are referring to a group of mice. So the researchers that were doing this really cool study was a couple that was studying neurobiology at Stanford University about 20 years ago. A married couple, two neuroscientists. Think about that. So they came up with this idea of testing whether or not old dogs can learn new tricks. Because everyone says you can. Holy cow, is that you, Nish?
Yep.
Is it okay? Okay. So old dogs, can you teach them new tricks? Well, some people say yes, and some people will flat out say no. You might be experiencing that with your parents right now, or your grandparents saying, oh no, I don't want to switch from my flip phone to a real phone. Oh no, don't tell me how to FaceTime. You know, they kind of give up. You know, I'm an old dog. I am not in the mood to learn new tricks. But can that happen? So these researchers said, okay, what we're going to do is we're going to have two populations of mice. We're going to have one population of mice that are in a cage, and they can eat all they want, all they want. We'll put an exercise wheel thing in there if they want to, but we're not gonna force them to exercise. So this is like the typical American, right? You're eating all you want. Oh, you know you should exercise, but most of us don't, right? So those kind of mice, they described as being not couch potatoes, but cage potatoes. An existence of not being very stimulated. You're living in a cage, but you can eat all you want. Then the other population of mice were put into a cage, but only temporarily. In this cage, instead of having an exercise wheel, they had an exercise wheel plus other things. And what they would put in a rotating basis, they would put toddler toys in their cage. So like rattles, things that made sounds or flashed light. So every week they had a different kind of environment that they had to kind of figure out, now what is this? Is this going to move? Does this sound scary? You know, in other words, they were being stimulated mentally on a weekly basis. And they were limited in terms of the amount of food that they were given, just enough. They weren't starving, but they weren't eating all they wanted. And for some reason, they took advantage, this is a very interesting thing, they took advantage of the exercise wheel, more so than the ones who were too full to move. You know, I guess if you're not eating very much, you have a lot of things to look at, you might want to exercise. Oh, that's interesting for humans. So what they found, these researchers, this married couple, was that after the mice died, they examined their brains and they looked specifically at the points of communication between neurons, which we call synapses. It's where one knob communicates with another nerve cell. That's the definition of a synapse. And what they found was that in the mice that were called cage potatoes, that the number of dendrites, you with me? The number of antennae? The number of dendrites actually got smaller and they retracted, which means that they're less likely to communicate with other nerve cells. So the number of synapses declined in those that were not mentally stimulated but could eat all they wanted and did very little exercise. I'll stop right there and say that is my dad 100%. My dad eats whatever he wants. He sits on the couch, he watches like Fox News and CNN and like John Wayne movies, cowboy movies. That's all he does all day, I think. You know, he doesn't move. And he's slower mentally. He can't keep up with the conversations at like birthday parties, and he's always like a step behind, you know? And what we find is that with the mice that are constantly stimulated, not only did they have the same number of synapses, the synapses were increasing based on their age. So they didn't kill mice. They said the mice could live until they died. And so some mice died at some ages and different ages. And they compared, they said, whoa, even long-lived mice still had an incredible number of synapses. So these mice were what we call better agers. They lived longer, they were mentally active, they were physically active, and they weren't overweight. And I think most of us should think of that and say, okay, that's a good role model for me, you know? Because some of us, I don't think any of you are close to thinking about retirement. I think a lot of you are thinking about, okay, what's my next move? But some of you have parents who might be approaching retirement, and you know what some people will say with retirement? I can't wait to stop working. I can't wait to get up like at noon, drink coffee, go play golf, eat dinner at 4 o'clock with all the old people, then go to bed at like 8, you know, whatever they do. What kind of existence? Well, that'd be fun for about a month, maybe. But then all of a sudden you have to start saying, how am I going to stimulate my brain? I can remember for my dad, I would say, after he retired, I said, Dad, you gotta do some nonprofit work, volunteer, do something, I think he did for a while, but his brain is retracting. So the point of this story is that when you slow down mentally, your dendrites and your synapses start to retract. That makes all aspects of your nervous system slower. I think you would describe successful agers, and some of you have that in your life, where you'll say, oh yeah, my grandmother, my mother, oh, she still is very active. She does a crossword puzzle every day. She's very active on her phone. She FaceTimes the grandkids, and she's very active, Yeah. Joanna?
Did they study at all whether it made a difference at what point in the mice's
Mhm.
life that they started the stimulation?
That's a good question. I think in this particular research study, they started them from birth. So it was always constantly giving them that chance. But what they found is that, even at different levels of age, the synapses would still continue to grow. So they were convinced that even really old mice were continuing to grow synapses. So that means that there's still opportunity to learn. And what the coolest thing about this research project was, when this couple, these neurobiologists, retired, this is the best, look, I'm getting goosebumps just thinking about it, is that they decided to practice what they preach. And for the most part, I would say that I try to do that when I teach. I try to, not give you suggestions without me saying, I'm trying to do it too. And sometimes I'll say, yeah, but that's hard. I realize it's really hard. But this is something that I hope all of us say we're gonna do. And that is, when they retired, they were Stanford neuroscientists. What a job. And when they retired, guess what they did as a couple? They learned a new language. Whoa, that's sweet. They each learned how to play a new musical instrument. Whoa. Can you imagine that? Age 75 or 65? Hey, let's learn a new language so we can go travel. Let's learn a new instrument. They even picked up sculpting. Neither of them were artists, but they were into, they were trying to increase the synapses. And I guess another example I have of this that's more kind of realistic for me was that at Xavier, I teach a class that I developed called the Biology of Aging. I love that class because it mixes anatomy with my interest in the process of getting older. And a couple years ago, I had a student in my class in her late 60s take my class. And that was a little awkward for me, because usually I'm like the old guy, Here I am telling them about my bifocals and stuff like that. And here I had a woman like almost 20 years older than me. And she sat in the front row, like right where Steph is, and she was giving me feedback the entire time. She was going like this, and she was smiling. She was writing things down. Just what I tell my kids to do when they go to college. I said, sit in the first two rows, make sure you give eye contact, make sure you smile, make sure you give them feedback that you're connecting. That's what people want to see. And then after her class, this is the best part, she rode her bike from Victory Parkway downtown, and she delivered soup for the soup kitchen on her bike. So she was taking a college level course at age 67, and she was physically active. She was like that mouse that was getting challenged physically and mentally. So there's a phrase that I hope you never forget, and it applies to many things in anatomy, but majorly when it comes to the brain, and that is use it or lose it. So if you have a parent who's approaching retirement, you might tell them about that research story. You might say, Dad, gosh, I know you enjoy your golf, but you got to find something that will continue to challenge yourself. Hey Dad, here's a computer. Why don't you learn how to do this? Or hey Dad, let's take a class together. Let's learn Spanish or something. That's gonna motivate me. You know, I obviously I tell this story a couple times a year, but that's gonna motivate me so that when I retire, my wife and I retire, we're definitely gonna do something like that. Physically, mentally, because so many people have the opposite kind of idea of what retirement is. Yeah, Michael?
Would you say that use it or lose it, but you can also, with a relative means, get it back?
Yeah, and that's the purpose of that research, is that you can still teach, you can still learn, you can still generate dendrites,
Yeah.
even if you take a break for a while. Yeah. So if you have a parent who's kind of in that mode, they can kind of start to sprout them again. So it is true that when you hear about that kind of phrase when you were in elementary school, hey, I learned something, I just grew a brain cell. Well, you didn't grow a brain cell, but you probably increased your synapses. You know, the more synapses, the better connections.
So do they just retract, or do they actually like die off?
They just retract, which means they no longer making physical, not contact, but they're not communicating anymore.
Okay.
They're still there.
Okay.
Yeah. So take a look at this screen. It says that with this number of synapses that we have, that, you know, there's maybe 800 synapses just in the spinal motor neurons. But when you get into the brain, there's so many of them. Synapses, connections, places of communication. So we said that in our kind of description of a neuron in lab is that when a neuron communicates with another neuron, we use the terms pre and post. So take a look at this diagram. This is kind of what I drew. I think mine was a little better than this drawing. You can see up on top we have one neuron communicating with another neuron. So the one that's releasing neurotransmitters is called the pre, and the one that's receiving those neurotransmitters is the post. So we can have different types of synapses, and they have names that look silly, maybe even confusing, but they make complete sense. For example, this presynaptic neuron is synapsing with, look, it's axon to the dendrites of the post. So we call it the axon and dendrite synapse. That makes sense. This one, the pre, is synapting with the axon and the cell body. And sometimes the cell body is called the soma. So we call it axosomatic synapse. And then this one is an axon and an axon. So it's an axo-axonic synapse. This one is kind of fun because this is the type of synapse that's generally utilized to block pain. So we have pain-producing cells. We have ways to naturally block pain, and it's usually by this type of synapse. Okay, so pre and post. Everybody's good, right? Now, you don't have to know the details of old Otto, but I think it's pretty interesting to talk about how he figured out what neurotransmitters are. It was around the time of the Civil War, and when people who were, you know, in, I guess, field surgery, they would maybe have issues associated with blood typing, and they learned all about blood typing and how blood worked for some people and not for others. But old Otto was a little bit before the Civil War, and he noticed that when he was looking at frog hearts, dissecting, that if he would take some juice from one heart of a frog and just squirt it on the frog next to him on the same dissecting pan, that heart could slow down or speed up. Something in the juice around the heart could control the rhythm of the heart. So we now know that what's in the juice was some chemical communicator. So remember what we talked about on Tuesday. We have an endocrine system which communicates through hormones, which is going to be the first thing that you learn in 152 in the fall. Or you have neurotransmitters. So hormones and neurotransmitters are the two forms of communication. In the case of heart, it has to do with neurotransmitters. So a neurotransmitter is something that can excite and the heart speed up or inhibit. Some neurotransmitters can do both. And some neurotransmitters, this gets a little confusing, can actually be hormones. So when you think of, for example, epinephrine, norepinephrine, those are usually considered adrenalines, you know. Those are hormones, but they can also act as neurotransmitters. So sometimes they can do both. Now the one that we talked about in terms of the muscular world was acetylcholine. Acetylcholine is typically an excitable type of neurotransmitter. Okay, so let's talk about the types of neurotransmitters, and remember this, oh my gosh, now see, I hope this doesn't sound mean, but for those of you who are here today, I'm looking around and you are here because you made it, right? Half the class is not here anymore. So you guys are saying, Mike, I can tell you all about this. These little bubbles, those are vesicles containing some type of neurotransmitter. In lab, we said it was acetylcholine. Here is the space, here is the sarcolemma, it's the neurotransmitter, or it's the postsynaptic membrane of the second nerve. So we're good with this, I hope. You could even pretend, the model looks just like what we're looking at. You can see exocytosis, the release of the neurotransmitter. So, and we see that, look, we see the receptors on the membrane. So everything is good. But what kind of response do we get when a neurotransmitter is released? Well, the response has everything to do with the type of neurotransmitter, and there's lots of types, and you do not have to study all of this information for the last test. But I want you to know a couple things about each category, and you can see that there is a category all to itself called acetylcholine. There's amino acid neurotransmitters, which you will hear about next semester when we talk about what influences the heart. There are some neurotransmitters that are derived from amino acids. And then, of course, you're going to hear all about neuropeptides with hormones and how they can influence and turn on and turn off. Yeah?
So for that frog experiment, those were the amino acid neurotransmitters?
Yes, amino acid neurotransmitters, yeah. And I think also they probably used acetylcholine too.
Okay.
Yeah.
So it could be multiple of them?
Oh, you can use them both, yeah, definitely. When we talk about mental illness, and this is not in your lecture outline, but just take a couple notes here. You don't have to write down everything. You know, mental illness is something that I think affects everyone in this room. Maybe it affects you personally. We'll even say things like depression, ADD, ADHD. That's a form of mental illness. We're going to say that. But I think all of us have friends, or there's that uncle, or there's that distant relative that has some serious problems. Oh my gosh. You know, without getting into detail, I could probably give you 10 examples of friends and family members who have mental illness that I know. 10, easily. And then just think about all of the kids that I've taught over 27 years. I had a student come to me this morning in my office at Xavier and said that I've missed the last 2 weeks of class. I mean, you can't miss 2 weeks in the summer. 2 weeks? I thought he was gone. And he told me he just had this mental collapse, you know? And I could see he was struggling to tell me about it. So when we talk about mental illness, we're talking about a disruption in brain chemistry. Oh, you've heard that before. But now what we're really going to be talking about in terms of a disruption is really the neurotransmitter that's being released. Some people will have a clinical diagnosis and they will say, oh, you know what, you are really low in dopamine. Or you're producing too much of whatever. So we're going to make a drug or give you a drug that either acts like the neurotransmitter that you're missing, or listen to this, or blocks it. Let's go back a slide. When we think of this diagram right here, this is how 90% of mental illness drugs work right here, just right here. And just think, you already can explain everything about this slide. So you can explain and understand how mental illness drugs work, and probably how street drugs work. Because when we look at these receptors and we say, oh, you do not have enough neurotransmitter, it could mean a problem with, look, exocytosis. Or it could be a problem with the receptor shape. And if you're producing too much, guess what the drug will do? It will try to mimic the shape of that specific neurotransmitter and block it, attach to the receptor and block it because your body's producing too much. So we have mental illnesses that result from over or hyper secretion as well as hypo secretion. Yeah.
So now this isn't obviously an every case type situation, but could you make an argument that there are certain mental illnesses that could be theoretically cured, I mean, via study, as an example? Like just basically intentionally keeping your mind active.
Yeah, yeah,
Prevents
no doubt.
a certain level.
100% yes. Yeah.
Diet and exercise.
Hold on.
I got a question.
Get over here.
What's the answer to tomorrow's test?
What's
I'm
going
picking
on,
A.
my brother?
I'm picking A.
What are you doing here? What are you doing here? Huh?
What are you doing? My wife is in her last,
I had this guy, what, 2 years ago?
yes,
And tell me, in this class.
yes.
Yeah, and this was the best news about it. You got like a D or C the first time, right?
I got a C. Don't
Yeah.
try to lower
All
it
right.
back.
You got a C, and then he took me again. The very same class.
Yeah, mhm.
And
Anatomy too.
and you made it. Now where are you at?
I had to set out nursing. My wife is pregnant. She'll be having.
Oh, okay.
Oh, did I tell you?
No, you're
Oh,
not.
okay. It's been a while. So,
Yeah.
you know, I was a nurse in 102.
Yeah.
Had this personal issues going on. Had to remove myself.
You don't have to tell everybody this, but.
Yeah, you know,
Yeah.
some people might be going through something right now.
Yeah.
Undisclosed,
Yeah.
and I'm not going into great detail, but
But you're on track.
yes, I'm back. I'll come back this fall
Okay.
to resume nursing
All right.
102,
Yeah.
and my wife is going to have your little baby girl.
Nice.
Okay, little baby girl.
That's her.
So precious.
He was one of the hardest workers. Yeah, and I don't think I've ever done this before, but after I had him for 3 semesters. So we were like brothers.
Yes, still
Yeah.
are brothers.
I took him out to lunch and I said, man,
Oh, do you still get an offer? If anybody get an A,
what?
they get a pizza.
A pizza?
The Roses. They can't go to our place.
All right.
Okay? You get the Roses, sorry.
Oh, wow.
I love you.
Good to see you,
Good to
brother.
see you.
Yeah.
Y'all can do it, all y'all A
Yeah.
students.
Oh.
Even if it doesn't reflect in your grades.
That made my week.
Yes.
I want. Good to see you, brother.
Yes, sir. I'll call you. I'll let you know when she has to get
when you
here.
become another father.
Yes.
All right. Man.
Should we get pizza?
I'm telling you, he works so hard, you know. And so for those of you who are getting a C, and just, oh man, he was one of my best students in my life. Yeah. All right, anyway, I can't remember what I was talking about. All right, so neurotransmitters. And Michael, you were asking me, ask that question again. I was
So it was just along the lines of it, like you can essentially through studying or just kind of keeping your mind active and learning, you can prevent certain, not all mental illness.
Well, some people believe so. Some people believe so, but if there is a environmental component, then that certainly would help. Some people believe that maybe a genetic issue would override that.
Yeah.
So, you know,
I think
So-
most people usually go along the lines of it just reducing the risk.
Yeah.
That
Yeah.
it was like.
well, not only being mentally active, but also physically active can have a lot to do with reducing risk. Take a look at some of these serious, I'm talking serious mental issues. So schizophrenia, oh man, that's a tough one. but it's all about producing too much neurotransmitter called dopamine. Dopamine is like the feel-good kind of neurotransmitter that's released in many cases with street drugs. So schizophrenia is just way too much dopamine. Look at this, depression, general anxiety disorder, ADHD. It has a problem with not enough norepinephrine and serotonin. Yeah.
So is that why those tend to run kind
Together.
of hand in hand?
Yeah, yeah. And then Alzheimer's, which is something we will talk about, is something that has a reduction in acetylcholine. Now, I'm not expecting you to memorize this, but the point again is that mental illness can be traced back in many cases to just a neurotransmitter problem. Look at these street drugs, cocaine. I mean, when you think about cocaine and you say, one of my favorite shows is like Cops. Cops Reloaded, yeah. So when I watch this show and they think that someone's on something like cocaine, they will automatically call for backup because this person is extremely confident and maybe like so confident that they think that he can take out like 4 or 5 people. That kind of confidence. Dopamine can boost that kind of ecstasy. But dopamine is something that's very short-lived, and so that's why it's so addictive, is that when you come down from that high, it puts you into a state of mind where you're saying, oh my gosh, right, I need that again. And then we can't probably go 2 days without hearing about somebody overdosing on heroin, and fentanyl, and oh my gosh. But interesting that I chose to put alcohol up here on this screen with heroin and cocaine. And the reason why I did that is because, and I talked about this a little bit on Tuesday, is that alcohol reduces the chances of synapses communicating by blocking it, ready? Because of hyperpolarization. Remember what we talked about at the beginning of class, that dip below resting membrane potential? That's what alcohol does. It causes a increased dip. So it's possible to respond, right? It's relatively unlikely, but it's probably not going to happen, and that's why reflexes and slurred speech and things like that continue to happen. And underline this abbreviation. This is the name of the neurotransmitter that's associated with alcohol, called GABA. So, neuropeptides, I'd like to talk about that a lot, but I'll save that for whoever you have for 152. Neuropeptides are neurotransmitters that usually cause something to linger longer. So we find neuropeptides being released in situations like digestion, believe it or not. When we want to trigger a continual squeeze or absorption or secretion during digestion, we have neuropeptides. Sometimes even athletes will take forms of neuropeptides to get a longer kind of response from some type of hormone that they're trying to use to give them benefit. They'll mix maybe human growth hormone with some neuropeptides, and it'll give them even a bigger boost. You'll hear. Hopefully you'll hear about that next semester. So we're gonna yes. Dave, I didn't even recognize you without the facial hair. Whoa, you look about 20 years younger.
I got a baby face.
What's up?
I was like, is that what they named Lance Armstrong for?
Oh my
the
gosh,
France?
100% yes.
Okay. Because he wasn't taking performance-enhancing drugs, but he was taking hormones, and they said...
He was taking every
Lectin.
hormone that your body naturally produces it, but he was taking it in excess. Yeah, if you would happen to take me for 152, eventually I have a question in my exam that says, tell me how Lance Armstrong cheated, because we talked exactly about that. I want you to know for the test, this will be a question, I want you to know about the 3 classifications of synapses. So we have a classification that involves something being excitatory. We call this a cholinergic, or a response using acetylcholine. We have a synapse that we just talked about, like the use of alcohol, that uses GABA, which inhibits. And then we have an excitatory synapse that uses a hormone called norepinephrine, which can act as a neurotransmitter. So acetylcholine, GABA, and norepinephrine. So it's 6:18. I thought I was gonna be finished by then. So over the next 10 minutes, we're gonna talk about these three classifications, and then we're gonna call it a day. Let's begin with a cholinergic. Now listen to that word, cholinergic type of synapse, which means it uses ACH, which you're all fine with because that's what we used with muscles. And look at it. You're looking at this and you're saying, Mike, I'm sick of this. Stop showing me this. Stop talking about the model. Stop talking about, look right here, everybody squint. Calcium rushing in, exocytosis, here we go again. Acetylcholine binding receptors. Oh, look at this. You know, action potentials. I mean, we're good. This was test 3, the whole thing. So we are good with an example of a cholinergic synapse because it is like a neuromuscular junction. Now, if you did poorly on test 3, and then you say, oh gosh, here we go again. So this is a situation where it benefits to kind of keep up. The opposite of an excitatory cholinergic response is one which causes and inhibits the response as a GABA response. And you can see that GABA stands for gamma aminobutyric acid. I would never ask you that, but that's what GABA stands for. And we said that GABA results in hyperpolarization. Hyperpolarization. So how does it cause the cell, this would be important for your answer on the test, how does it cause the cell to become hyperpolarized, which means even more negatively charged? It opens up, look at this, chloride gates. And so chloride, which is not very common in the extracellular fluid, comes in. And it makes the inside even more negative, which means, ready, if you're staying with me, it makes it even harder to reach threshold. Remember, once you reach that dotted line, you get the spike. But if you're hyperpolarized, it's even harder to reach threshold. That's why things are always delayed. So I have a quick video, about a minute long. I think it's really good, but the guy talks really fast. So this gives you kind of an idea about what GABA does with alcohol. Still can't believe Anwar showed up.
This is not another treadmill. Alcohol, one of the most commonly used and abused drugs in the world, comes in a variety of forms and does one thing quickly and well. It changes the way you feel. But to what do we owe these feelings of release and freedom? How could the ingestion of one simple substance so drastically change not only your actions, but thoughts and perception? As alcohol enters the body and makes its way to your brain, it begins to interact with your neurons or brain cells. In our previous video on marijuana, which you can click here, we explain the actions of neurons and their chemical messengers called neurotransmitters. But there are 2 main types of neurotransmitters that these neurons use to communicate or send electrical signals. Excitatory neurons use glutamate, while inhibitory neurons use GABA, the yin and yang, if you will. Much like the zeros and ones of a computer, these 2 messengers have contrasting roles. Glutamate begins a wave of excitation, while GABA not only inhibits this flow, but helps to organize and differentiate the results in your brain. And just like a computer, these signals can be combined in enormous complexity to form actions and thoughts. So how does alcohol affect these 2 signals? Turns out it suppresses glutamate transmission and enhances GABA transmission, so you get less excitation and more inhibition. And because glutamate sites become less effective, information flow becomes slow and only the largest signals can make it through. This means you feel less, perceive less, notice less, and remember less. In conjunction, the increased GABA hushes the excessive background noise of activity, fine-tuning and clarifying your thoughts, but to an excessive degree. This is why you may have heard that alcohol is a depressant, but inhibition of your neurons is not the same as social inhibition. Instead, it simply cleans up and removes extraneous activity by removing less significant excitation. Without normal GABA transmission, the excessive excitatory action would resemble an epileptic seizure. There would be no clarity of thought. While drunk, your GABA channels are wide open. Combined with the lack of glutamate, you begin to think very little, but with great clarity. It's what causes the momentous attitude of a drunk, often repeating the same idea or proclamation over and over, thinking clearly about almost nothing.
Is that on YouTube?
Yeah, it's on YouTube.
There's that last statement. Whenever I drink, it's highly political, religious conversations that are
Yeah.
pretty in depth, and it'd be awesome if I remember them.
Yeah.
But over and over.
Yeah,
It's on
over and over.
YouTube. It's called Your Brain on Drugs, and this same thing, they have it for every drug, cocaine, marijuana, everything. So, Your Brain on Drugs, alcohol. Okay, so we've talked about an excitatory, and glutamate would be an excitatory, like a cholinergic response. We talked about an inhibitory response, and now we get to talk about some crazy response using norepinephrine. Now, don't panic up here. Norepinephrine is an adrenaline. It's also a neurotransmitter. And what I want you to remember about norepinephrine, because we're going to talk about it more on Tuesday, is that it activates or communicates through a series of enzyme steps that results in making more and more enzymes. So whenever you see the phrase, and underline this is an NCLEX question, whenever you see the phrase second messenger, underline that, the result is that more enzymes are being made. So it's a many-step procedure. Sometimes they call it a cascade of events or multi-step pathway. That's what the second messenger system is. And I guess if this was biochemistry, we'd talk about cyclic AMP and all this stuff, but we're not going to. All I want you to remember from norepinephrine is that it excites, but it also makes a ton of enzymes as a result. And I'll talk about the significance of making more enzymes on Tuesday. So here are some of the steps. You can see that here's the cleft, that's this space right here. Here's the neurotransmitter, and then it has, look, numbers of steps, but the enzyme is that the result is that we're synthesizing, making a lot more enzymes. So the last slide of the day, we're doing okay, I think, is something that we've talked about already. What do we do when we have to get rid of those neurotransmitters? We want to stop the signal. We know that in some cases, like with acetylcholine, we have specialized enzymes. We talked about this with the sliding filament theory, something called ACHE. You might remember that, acetylcholinesterase, chewing up the residual acetylcholine. In some cases, you can see that, actually in most cases, the neurotransmitters are sucked back up into the knob by endocytosis. So we have endocytosis, we have acetylcholinesterase and other similar enzymes. And that is the main way that we can slow things down. Okay. All right, so 6:26. I told you we'd finish early, so yay, yay. Tuesday, we're going to pick up. Now, I know a couple of you maybe weren't here at the beginning of class, so let me reiterate. This is for a few of you, so just freeze tag. Next week will be our last week of lecture. So Tuesday and Thursday will be our last week of lecture. The following week, which I believe is August 6th, that Tuesday, is no lecture day, even though it says it on the syllabus, but it's a day in which, ready for this, all of you will have your lab final on that day, and we'll do kind of a shift thing that we did before. It's also August 6th, the day that you have to turn in your final exam. And now the final exam is going to be lengthened because the final exam that was supposed to be a sit-down on August 8th has been eliminated because I'm having this eye surgery thing. So here's my plea to you, that if you have not started this take-home, then start it this weekend and say, I'm gonna get 80% finished by Thursday. Otherwise, you know, this normally is like between like 8 and 10 pages typed. I'm adding another 15 or 20 questions on Thursday that will be due Tuesday. So you're having a test added on to a take-home test. But everything's take-home, and you can use your book, your computer, everything. You just can't use a person. So if you wait to the very end, it's gonna be overwhelming and your answers are gonna be crap. And then all of a sudden, what should have been an easy A, I think, could end up being a B or C. So I'm expecting detailed answers, not one-sentence answers, not even two-sentence answers. I want explanation. If I say you can use your book and you can use the computer, I want really good answers. So work on that this weekend. Work on it this weekend. Say, you know, I'm gonna get half of it done by Sunday night, and then maybe the other half by Thursday. And then you'll be ready to go. Yeah.
Does the rest of it go up after Thursday?
Yeah, I'll post it Thursday night,
Okay.
Thursday right after class.
I looked after class last time.
Yeah, it won't be posted until Thursday. Next Thursday, it'll say revised, and then the new ones will be added. So that's the plan. If you have lab tonight, since it's 6:30, let's say we start a little early. Let's